Before dinosaurs ever ruled the land, Earth's waters belonged to armored fish, giant sea scorpions, and predators built unlike anything alive today. But some of their most famous reconstructions are wrong. One legendary 30-foot monster probably never existed. Another animal long portrayed as a killer may have survived by feeding on plankton. And the lineage that endured the greatest extinction of all was not the biggest or the strongest. These are 9 prehistoric water monsters from the world before dinosaurs, and the extinction that changed everything. Number 9, the giant endocerids that ruled the Ordovician seas and the monster that grew too large for its own myth. In 2015, Christian Klug, a paleontologist at the University of Zurich, joined a team examining a question that had followed Paleozoic fossils for generations. How large did marine animals become 470 million years ago? This was a time when vertebrates were still small. Dinosaurs, mammals, and even the earliest four-legged animals remained hundreds of millions of years in the future. Among the largest known animals in those Ordovician seas were endocerids, cephalopods, with long, straight-chambered shells. Popular documentaries often gave the starring role to Endoceras, but the fossil behind the most reliable giant size estimate belongs to a different genus, Endoceras. Like other endocerids, it carried a rigid tapering shell divided into chambers that controlled buoyancy. Its soft body, head, and arms occupied the open end. Exactly how it swam and hunted remains uncertain because almost none of that soft anatomy was preserved. For decades, books and documentaries repeated a precise-sounding maximum length, 9 meters, 30 feet. A predator the size of a school bus patrolling the shallow seas of ancient Laurentia. But when researchers traced the evidence behind that number, they found no specimen that could confirm it. The 9-meter figure traces back to a 1955 account by the paleontologist Rousseau Flower. He reported hearing about a shell of that length from a New York quarry, supposedly destroyed before it could be preserved. There is no photograph specimen number or independently verified measurement. The largest well-documented endocerid shell is an incomplete specimen of Endoceras giganteum in Harvard's Museum of Comparative Zoology. It measures about 3 meters as preserved. Klug's team estimated its complete shell at 5.73 meters. Still enormous. Still among the largest known Paleozoic invertebrates, just not 30 feet, closer to 19. Yet the real mystery is not the missing 30-foot shell. It is the evolutionary pattern surrounding the confirmed giant. Klug's team was mapping something larger, a pattern in which cephalopods, trilobites, and eurypterids independently produced unusually large forms during a roughly 28 million year interval. That interval overlaps with the Great Ordovician biodiversification event, when marine diversity expanded dramatically. Several forces may have contributed, including new ecological interactions, changing food webs, and shifts in ocean conditions. No single mechanism explains every giant. The 9-meter shell remains an unverified story, but the evolutionary expansion that produced several very large animals in the same broad interval was real, and the confirmed evidence is strange enough without exaggeration. Somewhere in a Harvard collection sits 3 meters of chambered shell broken at both ends. It is the strongest physical evidence behind one of the largest animals known from the Ordovician Ocean. We cannot confidently reconstruct every detail of its body behavior or place in the food web, but we know it lived before jawed vertebrates became major competitors and long before the first dinosaur egg was ever laid. Whatever allowed endocerids to reach that scale was part of an ancient ocean we are still learning how to understand. Number 8. Ohio's spine-armed predator unearthed from the bucket of a steam shovel. In the summer of 1938, a University of Cincinnati student named Charles Cox was working on a highway crew near Manchester in Adams County, Ohio. The crew was cutting through Upper Ordovician limestone and shale. As a steam shovel turned the rock, Cox noticed a dark, textured fossil among the shell fragments. He pulled it free before the next scoop could bury it again and brought it to his geology professor, Kenneth Castor. Castor recognized what earlier collectors had misunderstood for decades. This was not plant material. It was an animal, and a strange one. In 1874, an earlier fragmentary piece of the same genus had been described by the geologist S. A. Miller. He mistook a compressed, spine-covered appendage for a graptolite, a colonial marine organism that resembles a plant. He named it accordingly, Megalograptus, meaning giant graptolite. The name remained even after Kenneth Castor and the paleontologist Eric Gelsvig Waring revised the genus in 1955. Using Cox's more complete Ohio material, they established that it was a eurypterid, a so-called sea scorpion, up to about 78 centimeters long. Its forward-facing limbs carried rows of hooked spines unlike anything on a modern arthropod. Fossilized droppings found in the same Ohio deposits contained fragments of trilobites and eurypterids, including material attributed to Megalograptus. Researchers have therefore suggested that this predator may sometimes have eaten members of its own kind. 78 centimeters does not sound large, but place it in the late Ordovician seas of 450 million years ago. Jawed vertebrates had not yet become major predators. Armored jawless fish did exist, but they were generally small. The most effective weapons in this ecosystem were claws, spines, crushing surfaces, and shells. Megalograptus carried a formidable set of them. The most unsettling clue did not come from its claws. It came from the coprolites. An animal eating its own kind is not by itself remarkable. Cannibalism appears throughout the animal kingdom for reasons ranging from opportunism to competition and food stress. What makes this case interesting is the context. Megalograptus shared its waters with much larger cephalopods and occupied a high predatory tier of its own ecosystem. The coprolites do not tell us why cannibalism may have occurred or how often. They simply preserve evidence that prey could include another eurypterid, possibly even another Megalograptus. That theme will return later. By the time we reach the final animal, fossilized waste will reveal an even more intimate form of predation. Charles Cox was not a paleontologist. He was a student on a summer job watching gravel move who happened to look at the right shovelful at the right moment in 1938. Nobody was searching for Megalograptus. It was simply waiting in the wall of a road cut, for the one person paying enough attention to notice that a piece of the Ordovician had been dug up and was about to be thrown away. Number 7, the sea scorpion, whose most dramatic journey survives only as footprints. In 2005, Martin White, a geologist at the University of Sheffield, examined an extraordinary pattern exposed near St. Andrews in Fife on the east coast of Scotland. 3 parallel rows of crescent-shaped impressions ran beside a deep, continuous central groove. They had been cut into what was once wet sand. It was unmistakably a trackway, and it was enormous. White published his findings in Nature later that year. The trail measured roughly 6 meters long and almost a meter wide. At the time, it was the largest walking trackway attributed to an invertebrate. White linked the pattern to a giant eurypterid called Hibbertopterus. This was a broad, heavy-bodied animal already known from fragmentary Carboniferous fossils in Scotland and elsewhere. Based on the track width and limb marks, the track maker was probably around 1.6 meters long. Some reconstructions allow a somewhat larger animal, but the exact size remains an estimate. Picture it. 330 million years ago, something roughly as long as a person drags itself across wet sand. Its limbs leave repeated crescent-shaped impressions. The rear of its body carves a central groove. Those 6 meters of movement will outlast everything else about the journey. For every Paleozoic arthropod known from body armor, there are others represented by what they left behind: burrows, borings, and in this case, footprints preserved in Fife. The trackway matters for more than its size. Eurypterids had long been considered capable of limited movement outside the water, but direct evidence from a giant individual was rare. White's trackway showed that a heavy eurypterid could support itself and move across exposed sediment under its own power. It does not reveal exactly how long the animal remained out of water or precisely how it breathed during the excursion. The colonization of land did not happen once in one triumphant moment. Different lineages moved across the boundary between water and land in different ways and at different times. By the Carboniferous vertebrates, and several arthropod groups were already established on land. Hibbertopterus was not joining them permanently. It probably returned to the water after crossing the exposed surface. But for a short stretch of time, it carried a body built for water across open sediment and left 6 meters of direct evidence behind. No body fossil has been recovered directly beside the Fife trackway. There is no complete animal to place in a museum case. There are only repeated limb marks and a central groove pressed into sediment that hardened into stone. The trackway gives us strong evidence that a giant eurypterid crossed this surface 330 million years ago. It cannot tell us why. Number 6, the armored giant that might not have been a predator at all. In 2020, a research team led by Samuel Cotham at the University of Bristol examined a nearly complete lower jaw from the southern Mader Basin in the Moroccan Sahara. Christian Klug was among the researchers, along with Jakob Winther and Emily Rayfield. The jaw belonged to Titanichthys, an armored placoderm that lived in late Devonian seas roughly 380 million years ago. It belonged to the broader arthrodire branch of placoderms, as did the predator Dunkleosteus, although the two were not members of the same family. For decades, popular reconstructions often treated Titanichthys as another armored killer scaled up to enormous size. The jaw told a different story. The team built digital models and ran them through finite element analysis, a stress testing method borrowed from mechanical engineering. They compared Titanichthys with Dunkleosteus and with another placoderm adapted for crushing hard prey. The results, published in Royal Society Open Science, strongly supported a different feeding style. Titanichthys had a long, narrow, toothless jaw that performed poorly under the stresses expected from biting or crushing hard prey. Titanichthys may have exceeded 5 meters in length, and its lower jaw could grow more than a meter long. But instead of crushing armored prey, the researchers concluded that it probably used continuous swimming and an open mouth to capture suspended food, somewhat like a modern basking shark. No filtering structure has been preserved, so the exact mechanism and prey remain uncertain. The jaw mechanics, however, make suspension feeding a strong interpretation. That reinterpretation turns Titanichthys into something more interesting than a mistaken monster. Very large suspension feeders evolved independently many times. The pattern appears in whale sharks, basking sharks, manta rays, baleen whales, several extinct bony fish, and even Cambrian arthropods related to anomalocaridids. These animals were separated by hundreds of millions of years and radically different body plans. Yet abundant tiny prey repeatedly made the same broad strategy possible: grow large, move through the water, and collect food that cannot fight back. Titanichthys wore the armor of a predator and was related to some of the Devonians' most formidable hunters. By appearance alone, it fit the role of a monster. The evidence now suggests something less violent, but no less remarkable: one of the earliest known giant vertebrate suspension feeders. For generations, its appearance encouraged reconstructions of a giant predator. A computer model of a jaw from the Sahara helped challenge that assumption. We do not know how many other fossil animals still carry identities built more from appearance than tested evidence. Number 5. The giant fish that turned Scotland's ancient waters into a hunting ground with few equals. In 1881, and again in a follow-up account 2 years later, a naturalist named Stock described an extraordinary find from Wardie on the outskirts of Edinburgh. A large flattened nodule contained a nearly complete fish. The head had been damaged by erosion from the Firth of Forth, but the preserved body stretched about 2.6 meters. It was among the most complete specimens then known of a fish, otherwise represented largely by scattered teeth scales and jaw fragments. The genus Rhizotus was named in 1840 by the anatomist Richard Owen. The species name Hibberti honors the Scottish geologist Samuel Hibbert, whose name is also preserved in Hibbertopterus. Later finds included isolated fangs reported at up to 22 centimeters long. Then, in 2018, researchers used CT scanning to examine a pelvic fin from the Wardie Shale. Inside was a robust bone comparable in position to a femur. It connected to 3 distinct supporting bones, rather than the 2-part arrangement familiar from tetrapod limbs. Estimates based on large jaws and other fragmentary remains suggest that Rhizotus hibberti may have reached 6 or perhaps 7 meters. A mass near 2,000 kilograms has also been proposed, although no complete giant specimen exists to confirm either figure. Even the lower estimates place it among the largest predatory fish, known from freshwater and deltaic environments. 330 million years ago, it hunted through the lakes, channels, and coal forest waterways of Carboniferous Scotland. Its size is impressive. The structure hidden inside its fin is more important. Rhizotus belonged to the rhizodonts, a group of lobe-finned fish on the tetrapod stem. In other words, they belong to the broader evolutionary branch that also contains every 4-legged vertebrate, including us. The CT scan pelvic fin is not literally a leg halfway completed. Evolution does not work toward a predetermined destination. What the fossil shows is that early members of the tetrapod branch experimented with several arrangements of robust internal fin bones while still fully aquatic. Those bones may have helped control the fin and transmit muscular force underwater. Related developmental systems were later modified in other lineages as limbs evolved. Rhizodus does not show a fish trying to walk. It shows that parts of the anatomical toolkit existed before walking became the job. Very few living fish approach these dimensions in freshwater. For a stretch of the Carboniferous in wetlands whose plant matter would later contribute to coal seams, Rhizodus occupied the upper levels of the food web. Then, environments changed and the rhizodonts disappeared. No direct modern equivalent inherited their exact combination of size, anatomy, and habitat. Number 4. The claw that revealed one of the largest arthropods ever known. In a quarry near the town of Prüm in western Germany, the amateur paleontologist Marcus Poschmann split open a slab and noticed a dark fossil trace. Careful preparation revealed part of an enormous claw-bearing appendage. Poschmann extracted the pieces, stabilized them, and reconstructed the structure. The complete claw was estimated at about 46 centimeters from base to tip, nearly a foot and a half. From one appendage, Poschmann brought the specimen to Simon Brady, a paleobiologist at the University of Bristol. Working with Eric Tetley, they published their analysis in Biology Letters in 2007. Using proportions from better-known Eurypterids, the team estimated a total body length of about 2.5 meters. Because the estimate depends on comparison with related animals, it carries uncertainty. Even so, Jacalopterus rhenanii remains one of the strongest candidates for the largest arthropod ever known. The fossil came from the early Devonian Wilwerath Lagerstätte, roughly 390 million years old. The environment was connected to river channels and marginal waters in what is now western Germany. Run the comparison plainly. This was an arthropod, a distant relative of modern spiders, scorpions, and horseshoe crabs, reconstructed at the length of a small car. Longer than most adults are tall. The claw also marks the high point of an entire vanished world order. Jacalopterus belonged to the pterygotids, a successful lineage of predatory Eurypterids. Other branches produced animals such as Megalograptus and Hibbertopterus, although those genera were not all close members of one single giant predator family. Eurypterids had already flourished for tens of millions of years. Then, aquatic ecosystems changed. Large, jawed fish diversified, and some were capable of treating Eurypterids as prey rather than rivals. Competition with fish may have contributed to the decline of giant predatory Eurypterids. Changes in habitat, oxygen, prey, and ecology may also have mattered. The fossil record does not identify one simple cause, and it does not prove that Jacalopterus stood at a hard mechanical limit for all exoskeletons. What the record does show is that no later Eurypterid currently known matches the estimated length of Jacalopterus. Eurypterids survived for more than 100 million additional years, but their final representatives were far smaller. For one interval in the early Devonian, something carrying a nearly half-meter claw moved through the waters of Western Europe. So far, no arthropod fossil found after it has convincingly surpassed its scale. Number 3. The armored fish, whose body plan vanished from the Earth. In 1867, a hotel owner and amateur fossil collector named J. Terrell was walking the Lake Erie shoreline with his son. Near the shale cliffs of Cove Beach in Sheffield Lake, Ohio, he found massive, curved plates of bone weathering out of the rock. Terrell had no formal training, but he knew he was not looking at anything ordinary. He called it informally the terrible fish. Decades of collecting followed. Peter Bungart of the Cleveland Museum of Natural History famously spent years preparing one specimen with tools that included a dental drill. The animal eventually became known as Dunkleosteus terrelli, honoring both David Dunkel and J. Terrell. In 2007, Philip Anderson and Mark Westneat built a biomechanical model of the skull. Their study identified a 4-bar linkage that could open the jaws rapidly and help draw prey toward sharpened jaw plates. Using the body size reconstruction accepted at the time, they modeled more than 4,400 newtons at the front of the jaws and more than 5,300 at the rear. Those values were model estimates, not direct measurements. More recent work has also challenged the traditional 6 to 8 meter body reconstructions. A 2023 study estimated typical adults near 3.4 meters, with the largest known individuals around 4.1 meters. Dunkleosteus was still a massive and powerfully built predator. It was simply shorter and probably deeper bodied than the classic school bus reconstruction. Dunkleosteus had no conventional teeth. Instead, sharpened bony jaw plates formed cutting edges that could be maintained through wear. They were capable of slicing flesh and damaging hard prey. fossils and bite marks are consistent with a diet that included other armored fish, early sharks, and ammonoids. Marks on Dunkleosteus armor also suggest attacks by members of its own species, although they cannot tell us whether every attack ended in cannibalism. Then the story moves beyond one predator. At the end of the Devonian, the recognizable placoderm body plan disappeared. In 2010, the paleontologists Lauren Sallin and Michael Coates examined vertebrate life across the Devonian Carboniferous boundary. Their analysis focused on the Hagenberg extinction about 359 million years ago. After that boundary, the distinctive placoderm lineages vanished from the fossil record. Their exact evolutionary relationship to living-jawed vertebrates remains an active area of research, and placoderms may represent several branches rather than one simple self-contained class. But no animal with the complete Dunkleosteus body plan survived into later ecosystems. The ecological roles left behind were filled by other groups. Sharks, ray-finned fish, and lobe-finned fish passed through the bottleneck and diversified in the Carboniferous. One surviving branch of lobe-finned fish eventually gave rise to tetrapods, and much later to us. Dunkleosteus was one of the dominant predators of the late Devonian, yet its form did not survive. The animal itself left no direct descendants that retained its armored design. Other less imposing fish lineages made it through. Their descendants inherited the emptied ecosystems. From the perspective of a giant predator, they may have looked insignificant. Evolution did not care. Survival, not dominance, determined what came next. Number 2, the amphibian that may have been the largest of its kind, reconstructed from frustratingly incomplete evidence. In 1948, the Brazilian paleontologist Llewellyn Ivor Price described a new genus of ancient amphibian from the Pedra de Fogo Formation. The fossils came from the Parnaíba Basin of northeastern Brazil across the modern states of Piauí and Maranhão. The holotype was fragmentary, including part of a skull estimated at roughly 50 centimeters long. Even so, Price recognized an elongated, sharp-toothed animal unlike anything then documented from South America. He named it Prionosuchus saw crocodile. Prionosuchus was not a crocodile. It was an Archogasaurian temnospondyl, part of an extinct amphibian-grade lineage. Long before true crocodilians evolved, these animals independently developed long snouts and an aquatic predatory lifestyle. The age of the Pedra de Fogo Formation has been revised over time, but current evidence generally places Prionosuchus in the early Permian roughly 278 million years ago. Further collecting produced several incomplete specimens. The largest, catalogued as BMNH R12005 and held in London, consists of highly fragmentary skull and postcranial material. Researchers estimated that its complete skull might have reached about 1.6 meters. The fossil itself is not a complete 1.6-meter skull. Some later reconstructions scaled that estimated skull to a body approaching 9 meters, creating the image of an amphibian as long as a city bus. That number became famous. It is not a direct measurement, and the original 1991 study did not provide a firm 9-meter total length. This is where the evidence demands caution. The giant estimate begins with damaged fragments, followed by an estimated skull length, followed by another estimate for the body. Comparisons with related archogasaurs and long-snouted living crocodilians suggest a total length above 5 meters may be plausible. 5.5 meters is a safer upper estimate often used today. 9 meters remains possible only under more generous assumptions. Prionosuchus was therefore one of the largest amphibian-grade animi. Prionosuchus was therefore one of the largest by a wide margin, goes beyond the evidence. The more complete temnospondyl Mastodonsaurus is another serious contender. The uncertainty is the point. We are trying to reconstruct the size limit of a vanished animal from several incomplete specimens, none of which preserves the entire body. Prionosuchus belonged to the Archogasaurs aquatic temnospondyls that occupied river and lagoon habitats before true crocodilians existed. Later reptiles eventually filled similar ecological roles, but they did not descend directly from Prionosuchus. The role returned. The animal holding it changed. In a London collection sits fragmentary material used to estimate a skull up to 1.6 meters long. It is not the only fossil evidence for Prionosuchus, but it is the main reason the animal became famous as a giant. The true maximum may have been larger or smaller than our preferred reconstruction. For now, its size remains an informed estimate, not a number carved into the fossil itself. The extinction that changed everything. Beginning in the late 1990s, the geologist Shuzhong Shen worked with geochronologist Samuel Bowring, paleontologist Douglas Irwin, and a broader international team to refine the timing of the end Permian crisis. Their work examined layered rock in southern China, including the famous Meishan section and the more expanded Penglaiton section. Thin bands of volcanic ash contained microscopic zircon crystals. Uranium lead dating of those crystals allowed researchers to place exceptionally precise ages around the extinction boundary. The results published across several studies placed the main marine extinction pulse at about 251.9 million years ago. A 2018 study of Penglaiton found no clear evidence of a gradual biological decline in the 30,000 years immediately before the main collapse. At that site, the extinction interval was estimated at roughly 31,000 years, with a similarly large margin of uncertainty. Other high-precision timelines place the main pulse within tens of thousands of years. That is not an instant in human terms. In geology, it is extremely fast. When the destruction ended, the marine world had been transformed. Traditional estimates place the loss as high as 96% of marine species. Some modern analyses produce lower figures closer to 81% after correcting for background extinction and gaps in the fossil record. Either estimate makes it the most severe known mass extinction of the Phanerozoic. On land, losses were also catastrophic, although exact percentages vary by dataset and environment. The crisis was more destructive than the asteroid-driven extinction that ended the age of non-avian dinosaurs roughly 186 million years later. Had conditions become even more severe, the lineages that eventually produced dinosaurs, mammals, and us might not have survived. This is the catastrophe toward which the entire story has been moving. Several animals in this countdown had already vanished before the end of the Permian. Dunkleosteus and Titanichthys disappeared near the close of the Devonian. Rhizodus vanished later in the Carboniferous. Jacalopterus was gone long before the final Eurypterids disappeared. Others belonged to lineages that reached the end Permian world. Eurypterids survived in greatly reduced form until close to the boundary. Archagosaurian temnospondyls also disappeared during the broader Permian crisis. A few xenocanth sharks relatives of the final animal on this list made it through. The main chain of events behind the Great Dying is now strongly supported. Massive Siberian traps volcanism released greenhouse gases. The planet warmed. Oceans lost oxygen and acidification added further stress. A Stanford-led study published in 2026 strengthened the link between heat tolerance, oxygen demand, and which marine groups survived. Researchers are still studying the relative contribution and timing of each stressor. The outcome is not in doubt. This was the most severe known biological crisis of the last half-billion years. Many groups that had defined Paleozoic oceans never recovered. Long-established communities collapsed and the survivors inherited a radically altered planet. But life did not stop. Number 1, the freshwater shark whose relatives survived the Great Dying and the disturbing evidence it left behind. In 2016, Odon Ogogain, then a PhD student at Trinity College Dublin, examined fossilized feces from the Minto coalfield in New Brunswick, Canada. The coal-bearing rocks are roughly 300 million years old and date to the Pennsylvanian subperiod. The coprolites formed tight corkscrew spirals, a shape produced by animals with a spiral valve in the intestine. Their size, shape, and the animals known from the formation led researchers to attribute them to Orthocanthus, a freshwater xenocanth first named by Louis Agassiz in 1836. O. Gauguin and his colleagues cut the coprolites into thin sections and examined them under magnification. Embedded inside were small three-cusped orthocanthus teeth belonging to juveniles. The team published the evidence in the journal Paleontology. The most likely interpretation was cannibalism. A large orthocanthus had consumed one or more juveniles of its own genus. The fossils cannot reveal whether this happened deliberately, how often it occurred across the species, or whether the adult was a parent of the juveniles. Orthocanthus may have reached about 3 meters in length. It had an eel-like body and teeth, with 2 large cusps and a smaller central cusp. For tens of millions of years, xenocanths occupied freshwater deltaic and occasionally brackish habitats across Pangaea. One possible scene is easy to imagine. Water levels fall during a dry season. A wetland breaks into smaller, crowded pools. Food becomes scarce, and a large predator consumes a juvenile of its own kind. That scene is plausible, but it is still a reconstruction. The coprolite preserves the meal, not the drought, the motive, or the relationship between predator and prey. The larger story is not simply that one shark-like fish ate another. It is what happened to the lineage when the world changed. Falkenlang and his colleagues were careful not to claim certainty about motive. Food stress in seasonal wetlands is one possibility. Opportunistic predation is another. The fossil cannot choose between them. Orthocanthus itself appears to have vanished during the Permian before the end-Permian mass extinction. But the broader Xenocanth lineage did something few groups in this story managed. Some of its smaller representatives crossed the Permian-Triassic boundary. While Eurypterids disappeared, and many temnospondyle groups were devastated, Xenocanths persisted into the early Triassic. They continued through the Middle Triassic and survived into the Late Triassic when the first dinosaurs already walked the land. Then the last known Xenocanths disappeared before the Triassic ended. The fossil record does not identify one sudden catastrophe as the cause. Their lineage simply fades from view. Eventually they stopped. Consider what that means beside everything else covered tonight. Giant endocerids vanished. Placoderms disappeared. Rhizodonts, archogosaurs, and eurypterids followed. Some were lost during mass extinctions. Others faded for reasons we cannot isolate. The lineage on this list that crossed the Great Dying was not the biggest or the strongest. Its survivors were smaller freshwater predators able to occupy habitats that most open ocean giants could never enter. Cannibalism may have helped Orthocanthus survive short periods of food stress, but the coprolite does not prove that this behavior carried Xenocanths through a later mass extinction. Their broader survival probably depended on several factors: body size, habitat physiology, diet, and luck. And in the end, they disappeared too, quietly, without one obvious geological event to mark the moment. Their disturbing feeding behavior came back to light only because corkscrew-shaped waste survived in a Canadian coalfield for 300 million years. You did not descend from Orthocanthus. Its lineage is not yours. But every direct ancestor you had across the Triassic, the Great Dying, and the Hagenberg extinction survived long enough to reproduce. Everyone. Without a break in the chain across hundreds of millions of years and repeated biological crises. Not because they were always the biggest, the strongest, or the animals the fossil record remembers first. Generation after generation, they were simply the ones that survived. Sometimes the margin was almost nothing. And you are what was left standing on the other side of all of it. If you want to see more videos like this, click the video on screen now and make sure to subscribe.